want to measure the concentration and purity of DNA or RNA, such as that you purified, like from a mini prep like we were talking about. A common way to do this is with UV spectroscopy, using something called Beer's law to calculate the concentration, and then looking at the ratio of the absorbance at different wavelengths in order to figure out the purity. Nanodrop, you have like a tiny little column, so you don't have to use very much sample, but it's the same principle as with the cuvette. So you're shining light through, and then you're measuring how much light makes it through. So not all the light's going to make it through, because some of the DNA and RNA are going to absorb some of the light. The more and more DNA or RNA molecules we have, the more light's going to be absorbed, and so Beer's law allows us to go directly between the two. Molecules can absorb light. So when we think about like colored things, like shirts, say, so part of the reason why you see it as this color is because it's absorbing all of the other colors. So white light is a combination of all of the colors of the visible light spectrum. And so you can think of the molecules in my shirt as kind of stealing some of those wavelengths. And so we can only see what's left over. And so we can think of the same thing happening in the ultraviolet spectrum. So with the rainbow, like Roy G. Biv, with the V being the violet. So if you go even past that, even higher frequency, you get to ultraviolet light. And we can't see this with our eyes, but molecules can interact with it, just like the molecules in my shirt can interact with the visible light. And so the molecules, a lot of like biological molecules, including DNA and RNA, they're absorbing light in this ultraviolet range. So they're absorbing this really high energy light. And so I'll talk more about this later, but it's because of the way that the bonds in these molecules are made up. And so they have to, they absorb, like they need more energy for those electrons to get excited, which is how things get like absorbed, like it's absorbed or whatever. But so basically they're going to absorb, they need this higher energy light to absorb. So they're not going to absorb the visible light much, but they will absorb this ultraviolet light. And we can take advantage of this to kind of like, it'd be like seeing what color my shirt is, but we're doing this with light that we can't see. And this allows us to measure the concentration of DNA or RNA. Or we can use this for other things as well with this thing called Beer's law, where we have an extinction coefficient, which is a measurement of how much a certain molecule will absorb at a certain wavelength. DNA and RNA, we can calculate these like rough, like the average values for these, as well as you can calculate the actual specific extinction coefficients, because they're going to depend in part on like how long your nucleic acid is in the sequence, because the different bases will absorb slightly differently. So I'll get into all of this later. Different molecules have different like favorite wavelengths, and this is typically where we measure their concentration, and like so we measure the concentration based on the extinction coefficient for that wavelength in that molecule. But different molecules, even if that's not their favorite, they might still absorb there to some extent. And this can make it seem like we have more of our molecule, which is why it's important to look at different ratios to see how pure our sample actually is. So instead of just looking at that one favorite wavelength where we get the concentration from, we also want to look at like a spectrum. So it's how it's absorbing other wavelengths of light and not just that single one. So we look at 260 to get, that's where we get the extinction coefficient. So you can see that the highest absorbance is 260. So you can see that you have an absorbance spectrum, so like a big curve, instead of just a single peak. That's because the absorbance is going to depend on like the electrons being excited. More about this in a minute. But basically different parts of the molecule will have electrons that'll can get excited by different wavelengths of light. And so within that same molecule, you can have absorbance at multiple wavelengths, and then there's some fuzzy like wiggle room with like vibrational levels and stuff as to how much energy those electrons need in order to be promoted to a higher energy state. Basically, the Beer's law says that the concentration, that the absorbance, so like how much light makes it through, or how much light doesn't make it through, depends on the concentration of the thing, the extinction coefficient of the thing, and the path length, so like how far the light has to travel. So Beer's law tells us that the absorbance, so how much of the light is going to get stolen as it's making its way through the cuvette or through that column of the nanodrop, is going to depend on three things. It's going to depend on that extinction coefficient, which we talked about and we'll talk much more about, which is an intrinsic property of the molecule. And so different molecules will have different extinction coefficients representing how well that molecule will absorb at that wavelength. And that's represented with this little curly E thing. We'll also have the concentration. So the concentration is going to affect this, because the more of the stealers you have, the more light can be stolen. So I don't have good props, but so if you take like this pipette tip, so you can think that if I shine light through here, it will encounter some of these and absorb. And the higher the extinction coefficient, the more each of these will absorb. Now imagine I have a full tip rack. Now it's going to encounter more of these, so it'll get intact more. And then the third thing is the path length. So you can think of, if I'm shining light through this way, it's going to encounter more of these than if I'm shining light through this way. And if I'm shining light through like two of them, then you're going to have an even longer wavelength. And so the more, it'll have more opportunities to hit. So even though these have the same concentration, so the same like density of their tips, if I extend the path length, I'm giving the light more opportunities to hit. So that's why the path length is going to matter. And so thankfully for us, most cuvettes are standardized to have a 1 centimeter path length, and like the nanodrop, it automatically calibrates things so that it's measuring as if it were a 1 centimeter wavelength, like when it gives you the values. So this is going to allow us to make some simplifications and take some shortcuts in our calculation using standard coefficients for DNA and RNA that are based on having a 1 centimeter path length. So you can get you can convert using a measurement that will give you like grams per microliter, just based on the, like it'll say the absorbance at 260 of single-stranded or double-stranded DNA will correspond to like 50 micrograms per microliter if you have an absorbance of 1. And so this is going to be like a shortcut that we can use to get an estimate of the concentration. Beer's law is going to let us get the concentration. That's assuming that your solution is pure, just that molecule. If you have other stuff in there that could also absorb, like proteins or salts, that could cause problems, which is why we're going to look at a couple of ratios of wavelengths and not just look at a single wavelength where we're doing the measurement based on the extinction coefficient at that wavelength. So basically, a spectroscoper, a spectrophotometer, I can never remember the correct terminology, but so there are kinds where you do it in a cuvette, kind of like that, or this nanodrop, which kind of, you put a drop here and put it down and make this little column. And what it's going to do is it's going to measure the wavelength, measure the absorbance of all these different wavelengths. Basically, a spectroscoper, a spectrophotometer, I can never remember the correct terminology, but so there are kinds where you do it in a cuvette, kind of like that, or this nanodrop, which kind of, you put a drop here and put it down and make this little column. And what it's going to do is it's going to measure the wavelength, measure the absorbance of all these different wavelengths. So it's going to shine light through. So basically with a nanodrop, you have like a tiny little column, so you don't have to use very much sample, but it's the same principle as with the cuvette. So you're shining light through, and then you're measuring how much light makes it through. So when we think about light, we typically think about like visible light, but that's only a small portion of the electromagnetic spectrum, or so what light is. So basically light is these little pockets of energy called photons, and packets of energy called photons, and they travel in waves. And the more energy those photons have, the faster those waves will travel, so the higher the frequency. And because they all have to travel at the speed of light, so they all have to get to the place at the same time, the ones with the higher frequency are going to have a shorter wavelength because they're going to have to go up and down more times before they get to the same place as a slow one. So the longer the wavelength, the lower the energy, and the the longer the wavelength, the lower the energy, and the lower the frequency. The higher the frequency, the shorter the wavelength, and the higher the energy of the photon. When I talk about like RNA and DNA absorbing UV light, well, UV light is itself like a spectrum. So we can look at specific wavelengths to get more information about the purity. This absorbance is at 250. Oh, it's 260, sorry. If you look, there shouldn't be much absorbance at 280. I mean, there's still some, but if you look at the ratio of 260 to 280, that's going to tell you about whether you have any protein contamination, because proteins most strongly absorb at 280. And we also look at 230. So 230 can tell us things about contamination from salts and phenol and things from if we were doing like a purification or an extraction. And it can also tell us about other things. And so we'll get into more of this later in a minute. But the key core principle is that by looking not just at that single value, but by looking at these ratios, we can get information about how pure our sample is. And then by using those extinction coefficients, we can get information about the concentration. And we can be really exact about this if we calculate like the exact extinction coefficients, or you can even get them. Sometimes, like if you order a primer or something, it'll be printed on there, or you can use like an online calculator. Or we can just use the generic extinction coefficients, which are typically translated into like these standard measurements. When we talk about like higher energy light, that's going to have a shorter wavelength. So it's going to be over on this, more towards this side. So you can see that the visible light, like violet light, is the most energetic. So it has the highest energy. Those photons have the highest energy. The wavelengths are the shortest, and the frequency is the highest. So they're going up, down more times per second. And at the other end, we have the red light. And so so it can get kind of confusing because increasing wavelength is going to correspond to lower frequency and lower energy. And our wavelength is going to be measured in nanometers, which are a billionth of a meter. Okay, so why am I telling you all of this? Because light's going to interact with molecules. So if we can represent, because we have these light in like different wavelengths have distinct energy of their photons. So because you have a direct relationship, remember, between energy and wavelength, so different wavelengths will have different amounts of energy in their photons. And so you can think of these kind of like baseballs being thrown. So in white light, you'd have all the colors, all these different baseballs of every energy. And then molecules called chromophores, chromophores are parts of molecules that can catch or absorb these photons. And as we'll see, what this is going to do is it can, if it's the perfect amount for that chromophore, it'll absorb that light. And different colors, different objects, different molecules will have different differences between like how much energy is needed to excite one of their electrons. And because light only comes in those exact sizes, it's kind of like if you had, instead of thinking about balls, you could think about like different denominations of money. So if you have like nickels and dimes and quarters and dollars flying at you, they'll have different prices that they'll want, those electrons will want. And so they'll only take specific ones, and different molecules will take different ones. And so this will give you information about the molecules. So these electrons. So basically electrons are one of the subatomic particles. So
elements come in atoms. So like carbons, hydrogen, oxygen, like individual atoms, like the smallest parts, units of them. They have um, even smaller parts within those atoms. So there's the protons, which are positively charged and hang out with the neutral neutrons in the central nucleus, which is the dense central core. And then what we're interested in here is the electrons, which are these negatively charged subatomic particles that fly around them. And the protons with the positive pole are tasked with helping rein in these negatively charged electrons. But the electrons would trying to get like away from the nucleus. And so the farther they are from the nucleus, the more energy, they need more energy to be further from the nucleus. If you imagine kind of just like having a bungee cord, when you're like pulling out and out, you need more energy in order to, takes more effort to get farther away. And so you can think of it kind of like that for electrons. And so if they absorb energy, then they can get to a more excited, higher, farther away state. And different molecules will do this the perfect amount. So remember, it takes the perfect amount to get between those levels. So these electrons are trying to pull away, but they can only exist at specific, distinct energy levels. And the difference between these levels is going to be different for different molecules. So they're going to want different wavelengths. And as well, like different parts of the same molecule will absorb different wavelengths as well. So it can get kind of complicated, but you can think of it kind of like a floor plan for like an apartment building. And you have these different floors, and then within the floors you can be on the bed, you can be on the table, you can be on the top of the dresser. And so how the distance between those different floors is going to be different depending on where you start. So this is why you get some fuzzy room and what's the favorite absorbed energy. And then the amount of energy you need to absorb. So the differences between these levels is going to be different for different molecules. And this thing called conjugation or electron delocalization, it's going to help decrease the difference between these levels. So it makes it easier for electrons to be absorbed. And this can push the absorbance into the visible light portion, because remember these require, these have a lower energy than this really energetic stuff. So typically molecules absorbing, like the transitions, the new energy needed are these really high amounts. So that's going to be really high frequency light, so really short wavelengths. But when you have that conjugation, so this like this sharing of electrons, this delocalization, such as in an aromatic ring, what that's going to do is it's going to decrease the difference between these levels and make it easier for the light to be absorbed. And so this is why you typically see conjugation in structures of like dyes and that sort of thing. So those often have really extensive conjugation. So you'll see like multiple ring systems where the electrons are being shared like all throughout all of them. But you'll also find just smaller bits of conjugation in common molecules. So when you have that when you have that extensive conjugation, then that can push things into the visible light spectrum. But we can also so typically molecules are absorbing a lot in things that we can't even see, even with our equipment, our spectroscopy, like our typical ones, like in the lab. But when you have those conjugation, if it's like a smaller amount of conjugation, you can push things into this ultraviolet range. And when you're in this ultraviolet range, we can measure that with our spectrophotometer. And so where will we find that? So in proteins and nucleic acids are two of the things that we commonly look at in terms of getting purity and concentration measurements from spectroscopy in biochemistry. And so proteins, they have these long chains of amino acids, and nucleic acids, so DNA and RNA, they have these chains of nucleotides. And so the places in those where you'll find this such conjugation are in these the aromatic rings of the bases of the nucleic acids and the aromatic protein rings of proteins. And then you'll also get some absorbance at this protein backbone. So this bond is resonance stabilized. This amide bond is resonance stabilized. And that's part of the reason why the protein backbone is so sturdy, but it's also, of course, causes it to have some absorbance at 230. So remember, you can have like different parts of the same molecule being absorbed, having different levels because you have lots of atoms in your molecule. And so then you can get a peak absorptions at different points. And we have to call these like lambda max, the peak points. So you can see that, so the backbone, you have, that's less conjugation than you have in like these aromatic rings. So it's going to, it's not going to push the energy levels down as close as much. So it's not going to close that gap as much. So you need higher energy light in order to see that. And remember, shorter wavelength is higher energy. And so that's why you get the backbone at this 230. And then these aromatic protein rings are going to be predominantly at 280. But what we care about most now is the nucleic acids in our talk. And so that's going to have the peak absorbance at EV 260. And also 230 is also, you'll get some absorbance from phenol, which can cause problems if you're doing an RNA extraction, but also salts. So when you're trying to like purify RNA or DNA, you want to make sure that to look at this 260 to 230 ratio in order to make sure that you don't have too much contamination from the salts and the phenol, because they'll absorb there. And so you also want to do a blank your spectroscopy. So make sure that whatever buffer or water, or whatever you're using that your sample is dissolved in, you want to test blank this blanket. So in the thing, so you say this is basically define your background and before you run your sample. So you don't get messed up. Okay, so we found the stealers. So remember, we have our light, we have our chromophores, we have the parts of that molecule that will absorb light. So you can think of DNA as having lots of bands like all over it, and that sort of thing. And so if we think about that, we have our catchers. And so for this, we'll just imagine we have like a single, like one DNA molecule will just have like one catcher, but remember it can have lots of catchers because you have lots of bases and also because you have different places on it. So you get slightly different shifted spectrum. But if we just use that simple catcher analogy, now we want to measure the absorbance, and we can do this with the help of the Beer-Lambert law. So if we think back to our light being those waves of traveling baseballs, and now we have our DNA or RNA or whatever the molecules in there are going to have different mix and absorb different wavelengths, as we talked about. And so that's what can be represented by this extinction coefficient, which tells you how well the molecule absorbs that wavelength. So if you have a higher molar extinction coefficient, or sometimes called like molar absorption, I'm not sure exactly if there's a difference technically in terms of some of the terminology, but the higher this epsilon is, the more each molecule is going to steal. So you can think of it as kind of like the probability that it's going to catch that molecule. So if you have a catcher that's really bad and the light can hit it, and then it still just drops the ball, like it doesn't catch the ball, you can think of it. So the better the catcher is, the higher the molar absorptivity. So the absorbance, remember, is like how much light's going through. or how much light's not going through. So how much of this light is being caught on its way through. And so the value, this is the value that the like spectrophotometer will give you. And this is going to depend on that extinction coefficient, as well as on the concentration and the path length. So the concentration, so how many molecules are in a given volume. So with a higher concentration, there's more molecules there to steal the light. And at a longer path length, the light's going to have to travel a further distance, and so it's going to encounter more of these molecules. So we have this this relationship, A equals the epsilon CL, so the absorbance equals the extinction coefficient times the concentration and the path. But what we want is that concentration. So we can rearrange this, and now we get C equals A for this thing. So 1 over extinction coefficient times the path length. And now we can take some shortcuts. So the path length, conveniently, is usually 1 centimeter. And even with the nano drop, it like adjusts it so it's as if it were 1 centimeter. And remember, so it might show it as like 10 millimeters, which is a centimeter. But so we can simplify, basically get rid of that L, because if we have a standard. But now we have this 1 over the extinction coefficient. And so now we have to get about what's the extinction coefficient. So it's going to depend on several things, including like what type of nucleic acid, and so we have different ones for like double-stranded, single-stranded, or DNA or RNA, as I'll get into in a second. But it's also going to depend on the composition, so there'll be different ones depending on the sequence, but they're all pretty similar. And so you can take like a generic average values for these, and these have been calculated to be these. And now you can then transform those into these standard coefficients. So if we go, if we divide the 1 over this part, then we end up with, sorry, this yellow box should be around this whole thing. So if we do that, we divide 1 by 0.02, we get 50 micrograms per mil, which is the same as nanograms per microliter. So this is telling you that if you have an absorbance of 260, that's going to, of 1, that's going to correspond to 50 micrograms per mil, or nanograms per microliter of pure double-stranded DNA. And if you had 2, that would be 100. And so for single-stranded DNA, honestly, like, I don't know why most sources say 33, because when 1 divided by 0.027 is actually 37. So this has really confused me, but some places say 37. So most places say 33, and I don't know why. And then RNA is 40. So if you had an absorbance of 260, 260 absorbance of 1 of RNA, that would correspond to 40 micrograms per mil. And so why is this? So that's really helpful, but so this is, so why these different values? So we have, remember I said the bases matter. So if you were to absorb all of these separately, you get that they absorb at slightly different extents. And so uracil is in RNA, but not DNA. And DNA has thymine instead. And thymine had uracil absorbs 260 better. So it's going to bump up the absorbance. And so this is coming into account. So sorry, I skipped a little. But if we want to think about purity, so we want to look at the 260-280 ratios, because remember, that's going to tell us about contamination from these other sources, because it's not just the DNA that's in there, and these other things can absorb some at 260 as well. But they absorb more strongly at different wavelengths, such as 230 or 280. And so but even when you're looking at those ratios, the ratios are going to be a little different for DNA and RNA, because they, so you might have noticed that the 260-280 ratio for pure RNA should be around 2, and pure double-stranded DNA should only be about 1.8. And this is because you have the uracil in the RNA, which has a much higher 260/280 ratio. And so that helps account for why you have different values for the double-stranded versus the single-stranded. And the reason why you have different values for the RNA versus DNA. And then if you have why you have different values for the double-stranded and single-stranded is because when you have this base pairing, it's actually, what it's doing is it's kind of like tying up those, the parts that normally catch the light. And it's kind of like changing some of the, those like energy levels and all sorts of stuff. And so basically you get this hyperchromic shift where it takes, so you see you have for single-stranded DNA, you have a higher absorption because those bases are free to absorb the light, whereas with double-stranded DNA, they're all tied up. And so this is going to correspond to the same like 1 OD. They often talk about like an absorbance of 1, like a OD is 260 of 1. So that means like if you measured the absorbance at 260 and it gave you a value of 1, that would correspond to 50 nanograms per microliter of double-stranded DNA. This should say single-stranded DNA, sorry. 33 of single-stranded DNA. And that's because each like gram or whatever of DNA is absorbing more than, or single-stranded is absorbing more than the corresponding double-stranded. And then one last note is that you also want to look at the absorbance at 320 nanomolar. So remember, now we're going a longer wavelength, so we're going to a more towards this visible spectrum, but not quite there. That is going to tell you about like turbidity. So it's going to tell you if like the sample is cloudy. And so you can actually adjust for this. Looking at the 260 minus 320 times your dilation factor, so how much you dilute the DNA, times the that little standard coefficient for the DNA. And you can find a lot more in my blog post, as well as if you are interested in the learning more about why things absorb, I highly recommend this like Chem Guide UK. This helps explain in more like detail where these changes in energy, like where these energy levels are coming from. And so where you can get this promotion of energy to these different levels and why you see it with like things with pi bonds. And so you're actually like, you can promote an electron from a pi bond into like an antibond, like a bonding orbital to an antibonding orbital. And then like the difference with more conjugation, you get the levels closer together. And so if you want the really geeky stuff about it, I really recommend this site. So